Hello everyone, welcome to the online session. In this session, we are going to discuss about ligand binding site prediction. We all know that ligand binding site prediction is an important step in the molecular docking and is to study of biological interactions of the molecules. So we're going to see how to predict the site using computational tools. There are many servers and tools are available developed for this for binding site prediction. So we're going to discuss one or two few tools today. Okay. So I'm going to share a screen now. Yeah. Here, hope it is visible. We can see here PDP data bank. Before going to study about the ligand binding site prediction, so first we need a protein file, protein molecules. Protein molecules file structures we can get it from protein data bank. So this three-dimensional coordinates of the structure can be downloaded from protein data bank because ligand binding site or active site of the enzymes can be predicted using some online tools. So here, these molecular structures or enzyme structures can be downloaded from protein data bank. So protein data bank, we all know that it's one of the major data bank. You can see here it is almost more than 1 lakh 50,000 molecule structures are predicted using NMR and X-ray structures. So you can see that NMR and X-ray structure, there are many different experimental methods they use it. So here I'm going to download on protein and also going to predict the active site region and the ligand binding site of the ligand binding site. You can see here, this is the protein which I prepared and chosen here, 6Y84. Either you can use a protein name or you can use the protein data bank ID, that means PDB ID. So in the search column, you can give a protein ID or a protein name of interest. On searching, you can get the number of sheets. You can also see that details here. So I'm going to download here SARS-CoV-2 main protease enzymes with unliganded active site here. So actually, they predicted already the active site region. So if you click on that pure protein data bank ID, you can see here the details of this particular protein. So that means the experimental details, the deposited, and their resolutions. There are many details you can get it, and also the literature you can also find here, and there are small molecule ligand. If any ligand molecules are already present, that means co-complex molecules present here. That also you can find out here. So, but we are going to download the structure of this particular protein. So we can easily identify the active site region in this. So how to download the structure here? So you can see here the right side in this column. So there are download files are there. You click on the different file format listed out here. So pasta sequence is there. Suppose if you want to sequence of this protein, you can download the protein sequence. And also if you want the protein structure, you can use this PDB format or zip format. So I need PDB format for active site prediction. So I just click on the PDB format. The structure will be downloaded here. 6 by 8 4 three-dimensional coordinates of structure downloaded here. So I'm going to open this. How to visualize this structure? Because to visualize this structure, we need specialized three-dimensional softwares. So here we have many softwares, PyMol, Chimera, VMD, but I'm going to use PyMol here. So you can see here, I hope it is visible. So you can see here the PyMol, I'm going to use it here. So you can see that along with this, there are some crystallography water molecule in the structure. Before going to predict active site of this protein, and let me talk about some of the information of this molecule, 3D coordinates. You can see here there are star molecules here. Those are water molecules because it's X-ray crystallography structure. So when they do a crystallography and X-ray diffractions, they use crystallography water molecules. If you want, you can eliminate this crystallography water molecules. You can see here by hide everything, hide waters. You can see hide waters. Now water molecule is gone. Suppose if you want to visualize in different model, cartoon model, you can also visualize in cartoon model. And if you want to hide a wireframe model, that also you can remove it. And also, if you want to find out the secondary structure element in the structure, you can highlighted with a different color. You can see here a structure structure element here. The helix is in a red color and the stands in a yellow color and loops in like green color. So this is a visualization tool. We can visualize it. We are going to visualize that. We can also use this software for visualizing active site region of the protein as well. Okay. So now I'm going to use the structure for active site prediction. So you can say I'm going to use, there are two tools. For example, yeah, so I'm going to use two tools. One is CastP server here. Yeah, CastP server here. So one of the best free available tool for this active site prediction, computer atlas of surface topography of protein. So that is that full form of CastP. So here you can click on the calculation part. You can see the calculations. After that, you can here you can upload the protein PDB file which we have downloaded. So you can see the protein data bank file we downloaded here 6Y84. It's a PDB ID. So I'm going to import that structure 6Y84. I'm going to import this structure and for access position. So and also you can use increase the active site size, the radius probe actually. So for example, active site, if you want, suppose your ligand molecules or drug molecule size is higher, the higher volume, then you can increase that active site according to that. So active site position, that it will predict the probe radius or radius probe or the active site. I mean, so for example, you can just sometimes you can use a default parameter. We can go to till 10 Armstrong, so degree. So you can see here 3.4, 1.4. So this is an email also you can give. It is an option. See here, and once it's uploaded, you can just submit it. Yeah. So after submitting, you can see here the mail as the link is given where you can check the results, active site details of this protein. So click on this web link. So it will take you to the another page. That means result page. So where you can get this active site of this protein details like this here. So you can see here the active site region here. So it is highlighted with surface model, and also you can see here the volume of the active site like 291 Armstrong, and the surface area also they given. Yeah. So these are the details you can. This is three-dimensional space. You can will be able to see this active site region. So also, if you want to see the show packet, there is different packets are already generated. For example, there are many packets. Here in this case, there are 36 packets are generated. If you want, you can choose second packet, or you can choose two, three packets sometimes. That means ligand binding site, and you can visualize it. So according to that, you can just check later which packet the ligand molecules can have a higher binding affinity. All those things you can just check here. Now I choose a packet one, and the packet one details are given here, amino acid details. For example, here you can see the sixth amino acid methionine is there, the active site. And after phenylalanine is there, proline is there, valine is there, aspartic acid is there, histidine is there, glutamine is there. All these amino acids which are present in the active site region is predicted and listed here. So you can use this active site as a ligand binding site parameter for further docking studies, any molecular interaction studies, because subset and inhibitors can bind into the active site, bind with the active site the region or allosteric regions. So you can use this molecule to check the interactions with the drug molecule and a subset molecule inhibitor molecule. So for the active site amino acids are very, very useful. So why actually you can choose entire protein, you can do blind docking, because sometimes computer takes more time to check binding interaction of any inhibitor or a subset molecule with the protein, because it has to check everywhere all the regions of the protein. Instead of that, if anywhere which if we can focus on the active site region, so that it will take less time to give a generated results, because it will check only the ligand binding interactions or a drug binding interactions, subset binding interactions with their own active site amino acids. So that is the reason why the people will do active site docking, protein dock, actually active site ligand docking. So it will reduce your time and it will give our exact results. So accuracy of the molecular interactions can be generated easily if you know the active site amino acid details. So this tool CASP Atlas, CASP Computer Atlas of Surface Topography Protein will give us this active site region. And one more tool I have here to explain you today, the active site. This is another tool, very important tool. You can also give a job ID like 6Y84. So job ID can be that, and you can also upload this protein file if you want. So downloaded protein file, you can upload it 6Y84. Yeah, you can see here I uploaded here. You can give your email ID, it's optional. If you want to change maybe in some protein, maybe change it, change B, change different domains you may have. Such case, which domain you are focusing, you can give it directly. So after that, find my binding site when you click. So it will just take you to the job is submitted to the server. You can see that. So how to check this status of the run, job run? You can go to the queue option here. Yeah. So you can just go to the queue option here. So you can see here when you click on the queue, yeah, you can see your status, your job is in computing queue now. So already some of the jobs are finished, sent by some other people, but your job is in computing, still it is in the queue. So let me show you all the already completed job here. So you can see here finished job it is. So you can see that you can download the PyMol session. This session can be visualized in the PyMol software. Yeah, this is how the active site regions it will look like. We can see here there are three active site. Active site will provide us the three best active site regions in the protein. So it will give us a rank order, the first and second and third. So you can always have that first site for it further any docking studies or any molecular interaction study. So yeah, these are the things you can just save this active site detail and you can use it for further docking studies, any molecular interaction studies, and further details. Yeah. I hope you enjoyed this session on identifying the ligand binding site in this. Thank you for listening.